Hello everybody! In this communication I will present the differential evolution between archaeal and bacterial communities in response to a Mediterranean extreme storm event. The presence and concentration of pollutants depend on land uses and on hydroclimatic conditions. We have previously demonstrated that an extreme storm event was at the origin of two peaks of multiple pollutants higher than ever along a year. They resulted from in-sewer solid resuspension during combined sewer overflows and runoff and sediments resuspension at the highest rivers flow. During this storm event, resident bacterial communities attached to suspended matter shifted in a pollutant-resistant and pathogenic community. Here I will present the Archaea evolution along the same extreme event and in comparison with how bacteria responded, to propose potential key players of pollution stress. To accomplish these objectives, water samples were taken during this autumn flood from T0 to T109 hours, in which environmental parameters were measured in parallel with the study of structural diversity of bacteria and archaea through metabarcoding sequencing. Both datasets were analyzed together through constrained multivariate analysis, and bioindicators of pollution were studied through network and modeling engine analysis. The archaeome was structured in 4 distinct communities along the flood event, as was the case of bacteria. Nevertheless, archaea behaved with delay at the highest flow that occurred at T38 hours, but its resilience was faster than bacteria. When linking abiotic and biotic information, the archaeome assemblages resulted significantly related to environmental parameters, while the bacteriome was further structured by environmental parameters. The exploration of potential bioindicators rendered one unique module significantly correlated to 2 environmental dynamics that comprise the parameters that peaked at CSO multipollution event for dynamic 2 and at T37 for diurean. The archaea within this module peaked therefore at the same moments. Archaeas known to be typical of sewage were found, as well as other taxa unknown to urban origin, and all of these taxa are potential bioindicators of water quality to explore further, as was the case of bacteria that I described in my oral presentation. Thank you for your attention. I'm open to your questions.